Right. Hi everyone, my name is Jing Zhang. I'm assistant professor in the horticulture department. I'm relatively new. I've been here for a year and a half. And so my program is plant phenomics. We focus on working with plant breeders to developing high throughput phenotyping tools to help them acquire more accurate phenotype data. And we also work with growers for some of the integrate some of the new cutting-edge technologies in precision agriculture to trying to map stresses in the field as well. So as you can see here, my poster just highlighted a few projects that we're currently on. I would like to really appreciate the NC Agriculture Foundation for funding our research and all the collaborators like a Sweetpotato breeder, Craig and Ken and Simon, and also the engineer Mike Kudanoff and the nematologist Adrian. So as you can see, we have this high throughput workflow specifically developed for the plant breeders from the canopy side and also the storage root side, so the two parts. Above ground, we're trying to monitor the growth using UAV images throughout the growing season. You can see it's a sample of different small plot sweet potatoes. And then we can extract a lot of different plant traits. And x-axis is the day after planting, y-axis is the canopy cover. As you can see, many different genotypes. They show great variations in different stages of their growth, and we highlighted a few lines on the standard checks, and the background is a lot of experimental breeding lines from the breeding program. And also that's the canopy, but we are really trying to link the pattern of the growth to eventually the yield. We also phenotype the yield. We fly a drone over the field. We have this image, and we extract them plot sizes, and we extracted the storage roots and analyzing the different traits. And we have a student, Alexis, working on this part. And for additional metrics, such as the sweet potato's length and sweet potato's width and the shapes. So, I forgot to mention we have another PhD student working on the canopy side. So they kind of like working together, and we'll try to tie those two components together and to better understand how the canopy changes and the growth differences contribute to the yield traits. Another project we are collaborating with Mike and Adrian is to try to detect if there's any signatures in remote sensing we can extract to indicate the nematode infestation in the sweet potato field. We collect some data in different dates. As you can see, there are two different dates. You have the visual picture, which is one single cultivar, and then we have a canopy temperature map that's showing differences. So usually hotter areas indicating some sort of stress. So you can see as the growing season goes, we can see a very, very significant differences in this one field. And this differences is very interesting to us, and it may or may not caused by nematode infestation. That's what we'll need to investigate later with collaboration with Adrian. So yeah, so that's just an overview of our research going on sweet potato. If you have any questions, let me know. So you can reach me at my email, jzhan256@ncsu.edu.
Hey, good afternoon, everyone. I'm Adrian Gorney. I'm the nematologist at NC State with responsibilities in sweet potato and row crops. So it was a really busy year for my team and I. We've been doing nematicide trials and collaborating with almost nearly every NC State researcher here on something related to guava root knot nematode. But this year we had 4 different nematicide trials, half on farm and half at our guava root knot nematode research site down at the Border Belt Station. We literally just harvested those trials, finished up this week. So I actually haven't yet had the chance to look at the numbers and analyze that data. But if you'd like to come to the production meetings that are going to be later this winter, I'll plan to present that information there. In these nematicide trials, we were testing about 20 different nematicides, including fumigants, conventional chemistries, biologicals, and some new experimentals that are in the pipeline. So a lot of diversity, really exciting stuff. We were also collaborating with Jonathan Schulteis's team and the Telios Ag team to look at a telone fumigant depth by rate study. So we were looking at two different application depths and two different rates, all in combination, to see what might be an optimal rate and depth for telone to control guava root knot nematode. We also just harvested that study, so I don't unfortunately have data to share with you, but I do plan to present that at our winter production meetings. We've also been collaborating with Jonathan Schulteis's team on these rotational crop studies, so looking at host and non-host rotational crops to put before sweet potato and how that impacts guava root knot nematode pressure in the subsequent sweet potato crop. Layering over the top of that some cover crops and fumigants as well. We harvested another data set on that rotational crop study this year and initiated 2 new trials. So we're keeping that rolling. Baker Stickley has been an awesome master's student keeping us organized on that and keeping that project up and rolling. Another project that my team and I have been involved with that we're very excited about is something that we're calling the NEMA AI project. This is a machine learning artificial intelligence project in collaboration with Dr. Edgar Lobaton at NC State. We are using images of the nematodes to train an automatic counting and detection system. So currently, when you submit a soil sample to the NCDA Nematode Assay Laboratory, they extract the live nematodes and they look at them one by one underneath the microscope. This is a really robust way of doing it. It's a really standard, gold standard way of doing it, but it does take a lot of time and it takes a lot of training of those technicians to do this. When they get thousands of soil samples, this can lead to a backlog. What we're hoping to do with this image analysis is to speed up that process and offload some of the work onto the computer so that it can identify and count those different nematodes. This is not to replace the technicians at the lab. We still need those trained technicians, but it is to lighten their workload and move through these samples a little faster. We just kicked off this project and we are focusing on root knot, soybean cyst, and lesion nematode at this time to train the models, but we hope in the future that this could be expanded to all of our different plant parasitic nematodes. So please check back for updates regarding that project. Lastly, I just wanted to kind of highlight some of the student projects on my team. So I'm very fortunate. I have 4 graduate students now, 2 masters and 2 PhDs. One is Michaela. She's a new master's student, and she is doing a survey of root knot nematodes in vegetables. So not necessarily focusing on sweet potato, but we're trying to get a sense of what are the major root knot nematode species that are impacting tomato and pepper and other vegetables across the state. A survey like this has not been done in North Carolina for about 30 years, so we really need to update our knowledge about what's affecting these vegetables so that we can address that. So Michaela has been working very diligently on that project. Another student, Jaron, down here in the front, he's working on guava root knot nematode in sweet potato. He has a really interesting project. We're looking at hot water treatment of storage roots as a method of killing the nematodes in that root. This is a very new area of research. Nobody's really looked at this before. And it's kind of a high-risk, high-reward project. We don't quite know what we're going to find, but Jaron's finding some really interesting things. And he actually has a poster here. It's the first poster just on the other side. PhD student Sarah, she is working in guava root knot nematode in soybean. So in her project, she's looking at resistance, trying to find resistance in soybean and working with USDA breeders in Raleigh to try to identify that resistance and further characterize that in soybean. She's also spearheading a project looking at extracting DNA of nematodes directly from a soil sample so that we can screen for guava root knot directly from the soil. It's a little bit technical, but it is tricky to get DNA out of soil and usable DNA. But Sarah has made some significant strides with that, and now we are pushing that a bit further, expanding to other species, and trying to push the limits, the lower limits of sensitivity for that test. And lastly, Mariah is a newer PhD student on my team, and she's kind of working across different commodities, but focusing more on the biology of the nematode. So looking at how the nematode may be moving in sweet potato fields, looking at potentially some resistance in tobacco, and a few other projects, but focused more on the nematode side of things. So I'm really fortunate to work with these great students and great collaborators all the way around. That is all the updates I had for you today. If there's any questions, or I'll be at the front table here if there are any questions.
Well, this is my first field day for any crop. I recently joined NC State just 3 months ago to run the Genome Editing Center for Sustainable Agriculture. It's exciting to be here and hear the different presentations and just see the care and the love that the researchers at NC State and in the NCDA and in the extension networks have for this crop, and the passion that the researchers have, as well as the interactions with the producers and the farmers. I would like to join your community, and I'd like to introduce myself a little bit and talk about what we can do at the Genome Editing Center to support the future of this crop in this great state. So my background before coming to NC State is I spent 13 years at Syngenta in our biotech division, which our headquarters are actually right by the airport, by RDU. And so those 13 years, I worked on improving crop genetics with new technology. That included working on GMOs and then new technologies, including this thing called genome editing, which I'll introduce to you today. Now, most of my work was in corn and soybeans and wheat and major row crops, but there has traditionally been a blocker for those types of technologies to be used in some of the more smaller horticultural crops, such as sweet potato. And so with this opportunity to start the Genome Editing Center, the real opportunity is to really quickly improve the genetics of some of these varieties using some new technologies. And I know for some folks in the room it might sound like these are GMOs. And so I want to just talk a little bit about the differences between traditional breeding, GMOs, and genome editing, because there's some really important distinctions there. But before I get into that, I'll just say my background, why I got passionate about this is I was a college student and I was studying biology and chemistry, and I really wasn't finding myself very happy with any of the topics I was working on, and I decided to take a year off from college and go work on farms across Pennsylvania and New York. Now, I didn't have a farming background, but I just thought it would be something good to do to find some, to kind of calm my senses, I suppose. And working on the farms for that year, I just really fell in love with plants and with agriculture. And most importantly, I got to see how incredibly challenging it is on a day-to-day level of just doing the basics out there. And having that kind of hands-on experience really inspired my passion to say, I want to really make this as easy as possible on farmers. And the thing that I thought would be the most important thing I could do is to work on the genetics. Because if you can get the genetics right, then you don't have to do extra work on spraying. You don't have to do extra work on scouting or other downstream stuff. And so that's the power of genetics is something that really inspired me, and that's where I tried to spend my career. And so I'm with the topic of future-proofing Covington sweet potatoes. We are really building on the great work by Simon, by Craig, by Adrian, and by many other colleagues who have discovered this resistance that you saw out there in the buckets on those 4 varieties on the right. And so that resistance exists in sweet potatoes, but it is really hard to breed it into the commercial varieties that we want to grow. And that's because of the complex genetics of sweet potato and just the long timelines and selection processes. You heard Simon talk about, well, I'm the guy who makes hundreds and thousands of lines so that we can then select them. And that has to happen year after year after year. And things are getting easier with the use of genetic markers. But the technology that I'm working on offers a shortcut through all of that. And so what we are going to start to do in the next year is take the gene, that one major effect gene that Simon was talking about, and take that gene from the varieties that have the resistance and put it directly into new varieties, including eventually in the next couple years, putting it directly into Covington. And the way that we do that is a lab step, okay? So this is not traditional breeding. There is actually a lab process that we use to take that gene and put it in. But what the outcome of it could be is that in maybe 3 years, we can have a Covington that has the resistance gene, and you haven't changed anything else about the line. If you do traditional breeding, as Ken was referring to, you get all this diversity of outcomes that you may not want, changing the quality, changing the flavor, changing the look, changing the response to other diseases. Well, if you can just put that one resistance gene directly in, you don't change any of that other stuff. And so now it does sound, what I just said probably, again, sounds a lot like GMOs, but there's a really important distinction here. So first of all, there's 2 things that GMOs are that this technology is not. The first is that GMOs use foreign DNA. By foreign, I mean not coming from sweet potato, not coming from the crop, okay? And so in this process that we're talking about using genome editing, there is no foreign DNA because we're just taking the gene that exists in sweet potatoes and putting it into another line. The second distinction is that GMOs, you can't make them through traditional breeding. They are impossible to get through traditional breeding processes. But what we're doing is basically the same that you would get through breeding. We're just getting there a lot faster, because I'm pretty sure if Craig and Simon live to and work on this for 20 years with Adrian and others, they could eventually get to a Covington that is almost identical to the current Covington with that gene. It would just take a really long time. We can get there in the lab in 3 or 4 years, then it's the same outcome as breeding, we just got there faster. And so at the molecular level, at the genetic level, if there's no foreign DNA and it's the same thing you can get by breeding, then it's not GMO. And that's according to the regulatory policies in the US, in the UK, in Canada, in Brazil, in Argentina, and hopefully in the next couple years in Europe. Now that's the thing we're going to be watching most closely, right? As we develop this project, we want to make sure and see how does Europe deal with this new technology. They do have a recommendation on the table that the EU Parliament is discussing and the member states are discussing that would put this type of technology in the non-GMO category, which is what we want, right? And so I would just like to wrap up, and I'm happy to take your questions. I would like to wrap up by saying that this is one of several projects that we're looking at, but I just met with our Genome Editing Center Council yesterday, which is a group of internal NC State advisors and external advisors that are helping us pick the right projects. And when we talked about this one, they ranked this right at the top, and they said this is important topic, the nematode problem is real, and we need to come up with a bunch of different solutions. Yes, we will have traditional breeding. Yes, you see those NC09 out there that looks good, but we want to have all the alternative options. And if we can get this to work for this one, what other things can we do in the future? And that's the other thing, is that when we build the path, we'll use this guava root knot nematode gene as our example. But even if we don't take it all the way in the next few years, that path will be paved and we'll be able to do other stuff in the future. And I was really happy to hear the council tell me that that's the one they thought we should work on. And so, again, I'd just like to say, in the next couple years, I hope that we can have some sweet potatoes to show you where we've done some stuff like this, where maybe even put it out in the field in the next couple years, and so you can see what we're talking about. And with that, I want to say thanks and enjoy the dinner. And if you have any quick questions, I can take them, or you can catch me after.